This is chapter 11. We're going to go over the nervous tissue. So in this chapter, we're going to look at the nervous system, how it keeps its controlled conditions within limits to help maintain homeostasis and also maintain your health. We're going to look at the different branches of the nervous system and also look at the different types of cells that are found within nervous tissue. With a mass of only 4.5 pounds and about 3% of total body weight, the nervous system is one of the smallest yet most complex of the 11 systems of the body. The central nervous system, abbreviated CNS, consists of the brain and the spinal cord. The spinal cord is connected to the brain through the foramen magnum of the occipital bone, and it's encircled by the bones of the vertebral column or vertebral column. The peripheral nervous system, abbreviated PNS, consists of cranial nerves, spinal nerves, and several other things that we will talk about a little bit later. A couple terms that we should know right off the hop is a nerve. A nerve is a bundle of hundreds to even thousands of axons plus the associated connective tissue and blood vessels. So this is a bundle of axons. Another term we will come across is ganglia. Now, ganglia are small masses of nervous tissue consisting mostly of neuronal cell bodies that are located outside the brain and spinal cord. So these are going to be located in the PNS, the peripheral nervous system. So we would define ganglia as masses of, well, it would be neuronal cell bodies, not just any cell bodies. In the peripheral nervous system. Now, the nervous system overall is broken up into the peripheral and central nervous systems. The central nervous system, like I said, abbreviated CNS, consists of the brain and the spinal cord. And then the peripheral nervous system, abbreviated PNS, is pretty much made up or makes up all the tissue outside of the central nervous system. Now it's further broken up into 3 other nervous systems. These are going to be the somatic nervous system, the autonomic nervous system, and the enteric nervous system. So just further divided the peripheral nervous system. Now, the somatic nervous system first consists of sensory neurons that carry information to the central nervous system from somatic receptors in the head, body wall, and limbs, and from receptors for special senses for vision, hearing, taste, and smell. So these are going to be special sensory receptors and somatic sensory receptors. In the somatic nervous system, we also do have motor neurons, and these are going to conduct impulses from the central nervous system to skeletal muscles only. The autonomic nervous system, abbreviated ANS, this consists of sensory neurons that convey information to the central nervous system from autonomic receptors. Things like breathing rate or heart rate, things that we don't consciously perceive really, are going to be sent by the autonomic nervous system. And the autonomic nervous system also consists of motor neurons for two different divisions. So the autonomic nervous system is further divided into the sympathetic and parasympathetic divisions. And these control smooth muscle, cardiac muscle, and glands. Lastly, then, we have the enteric nervous system. This is also known as the brain of the gut. And this is completely an involuntary nervous system. Now, the ENS consists of over 100 million neurons in the enteric plexuses, and these are going to extend through most of the length of the gastrointestinal tract. So the enteric nervous system is really the nervous system all around the gastrointestinal tract. Here we have the different types of neurons within the nervous system. So first we have sensory neurons. Sensory receptors will detect internal stimuli such as an increase in blood pressure, or sensory receptors attached to sensory neurons will also detect external stimuli like rain dropping on your arm. So this sensory information is then carried to the brain and the spinal cord through cranial and spinal nerves. There's also going to be motor neurons that send information from the central nervous system to the peripheral nervous system to respond to the stimuli that is sensed by sensory neurons. And then we also have integrative neurons. Now, the nervous system processes sensory information by analyzing it and making decisions for appropriate responses. So this is an activity known as integration. So these integrative neurons are going to be the neurons that process and analyze the information coming in. Help us to make decisions, or help the neurons to make decisions in the very least. Now, once sensory information is integrated, the nervous system may elicit an appropriate motor response by activating effectors. So these are going to be things like muscles, glands, that kind of thing. And these are activated by motor neurons. So let's look at a little bit of histology. We're going to look at the two main cell types within the nervous system, neurons and neuroglia. So the nervous tissue consists of neurons and neuroglia. Now, like muscle cells, nerve cells, also called neurons, these possess electrical excitability. Now, this is the ability to respond to a stimulus and convert it into an action potential. So remember, a stimulus, this is going to be any change in the environment that's strong enough to initiate an action potential. An action potential, remember, sometimes abbreviated AP, or also known as a nerve impulse in the case of nerve cells. This is going to be an electrical signal that propagates or travels along the surface of the membrane of the neuron. Now, it begins and travels due to the movement of ions, specifically sodium and potassium. So going to be the two main ions that are involved in the movement or the propagation of an action potential, and sodium and potassium move between interstitial fluid and the inside of the neuron through very specific ion channels in the plasma membrane of the neuron. So here we have a neuron on the right. Let's kind of just look at some of the structures of a neuron here. So here we have the cell body. This is just going to be this entire area here. This is the whole cell body. And the cell body is also called a soma. So if you see soma in your text anywhere or anything like that, it's just another term for cell body. And the cell body contains a nucleus surrounded by cytoplasm, like we see in other cells. And the cytoplasm is going to contain those typical organelles that we know from other cells. Now, cell bodies also contain free ribosomes and really prominent clusters of rough endoplasmic reticulum that we specifically call Nissl bodies. So Nissl bodies, these are going to be clusters of rough endoplasmic reticulum. Nissl bodies actually give the cell body of neurons a gray-colored appearance. Now, the cytoskeleton includes neural fibrils as well as microtubules. So neural fibrils, this is composed of bundles of intermediate filaments, and they help to provide the cell shape and support. So we'll add this in here. Okay. We're also going to see microtubules within the cell body, and these are going to assist in moving materials between the cell body and the axon, which is this long portion of the cell. Now, a nerve fiber, this is going to be a general term that we really use for any neuronal process or any extension that emerges from the cell body of a neuron. So most neurons will have two kinds of processes. They're going to have multiple dendrites, which are labeled up here, attached to the cell body, and a single axon. So often they'll have lots of dendrites, but only one axon. Dendrites are going to be the receiving or input portion of a neuron. Dendrites are usually very short, tapered, and quite highly branched, and we can definitely see that here. A single axon of a neuron is going to propagate nerve impulses towards another neuron, sometimes even to a muscle fiber or a gland cell. The axon is a really long, thin, cylindrical projection that often joins to the cell body at a cone-shaped elevation, which is called the axon hillock. So this is going to be the connection point between the axon and the cell body, that axon hillock. Now, the part of the axon that's closest to the axon hillock is called the initial segment. So the initial segment's part of the axon. And in most neurons, nerve impulses, otherwise known as action potentials, will arise at the junction of the axon hillock and the initial segment. This is an area that we call the trigger zone. So this is at the junction junction of the axon hillock, and initial segment. This is going to be called the trigger zone. And remember that trigger zone is where the nerve impulse initiates, where it starts. And it's going to travel down the axon from that trigger zone. Now this action potential is going to travel down the axon till it gets to the axon terminal, and then to the synaptic end bulb, where there's going to be another cell here. Could be a neuron, could be a muscle cell or a gland. For example, here I could just see it's a muscle, where that synaptic end bulb is going to make contact with. And at this point where there's contact, this is going to be the site of communication between the neuron and the muscle cell. This could also be a site of communication between two neurons. Remember, this area, there's going to be a gap between them, and it's called the synapse. Remember that synapse is that site of communication between a neuron and its effector. Both structural and functional features are used to classify various neurons, just like the classification of joints that we looked at in previous chapters. So we have multipolar, bipolar, and unipolar, and this depends on how many neuronal processes they have. Most neurons of the brain and spinal cord are going to be multipolar. Also, motor neurons are multipolar, so this means that they have multiple neuronal processes, always having one axon, but they have many dendrites. Bipolar neurons, these are found in the retina of the eye. Mainly, they're also found in the inner ear and the olfactory or smell area of the brain as well. So they will have one axon and one dendrite, bipolar. And then there are unipolar neurons. They usually begin in the embryo as bipolar neurons, but then as they develop, they become unipolar. During development, the dendrite and the axon will actually kind of fuse together and become a single process off of the cell body. The dendrites of most unipolar neurons function as sensory receptors. So these are almost always sensory receptors. Now, in addition to structural classification, some neurons are going to be named for the histologist that first described them. So Purkinje cells, these are found in the cerebellum. And pyramidal cells, these are found in the cerebral cortex of the brain. So Purkinje cells are named after the histologist that discovered them, whereas pyramidal cells are named because they kind of look like a pyramid. So they're just more named after their shape. So those are the structural classifications, the bipolar, unipolar, and multipolar, and then Purkinje and pyramidal cells. And then here we have the functional classification of neurons. So we have sensory neurons. These are also called afferent neurons. These are going to carry information from sensory receptors to the central nervous system. So they carry information from sensory receptors to the central nervous system. Motor neurons are also called efferent neurons. These send information away from the central nervous system to effectors, usually which are muscles or glands. So information from the central nervous system to effectors. Muscles or glands, usually. And then we have interneurons. Interneurons are also called association neurons. And these are mainly located within the central nervous system between sensory and motor neurons. Interneurons will integrate incoming sensory information. Integrate, another word for integrate is to kind of process this incoming sensory information from the sensory neurons. And then they will help to elicit a motor response by activating the appropriate motor neurons. So interneurons are kind of the processing and decision neurons in very, very simple, like 3 neuron systems. So we have like a sensory neuron connected to an interneuron connected to a motor neuron. So this is just a very, very simple 3 neuron system. In this case, the interneuron is going to be the processing neuron. Most pathways are going to be a lot more complex than just sensory, interneuron, motor neuron, but this is just the simplest form here. Most interneurons are going to be multipolar in structure. Now these are examples of sensory receptors that are dendrites of unipolar neurons. One thing of note here, these little breaks, that little circle here, these little breaks in the picture, these just indicate that the axons are going to be a lot longer than are shown in the picture. So just a quick note there. Okay, so first off here we have a corpuscle of touch. This is a touch receptor that consists of this mass of dendrites enclosed by a capsule of connective tissue. Then we have type 1 cutaneous mechanoreceptors. This is a touch receptor that consists of these free nerve endings, which essentially are bare dendrites, and they make contact with tactile epithelial cells of the stratum basale in the skin. So these are just free nerve endings. Whereas this is encapsulated nerve endings. They're enclosed by a capsule. Nerve endings, or I guess dendrite endings. Dendrites, you could also say, or free dendrites. Then we have a lamellated corpuscle. This is a pressure receptor. It's composed of a multi-layered connective tissue capsule that encloses a dendrite. And then we have a nociceptor. This is a pain receptor that consists of free nerve endings as well. So this one is a pain receptor. This is a pressure receptor. These corpuscles of touch, remember these are also called Meissner's corpuscles. And these detect usually deep vibration and pressure. And these type 1 cutaneous mechanoreceptors will detect more shallow kind of touch, pressure, and vibration. or fine touch. Moving on to neuroglia. Now, neuroglia make up about half of the volume of the nervous system. Now, neuroglia are a lot smaller than neurons, but they are 5 to 25 more times more numerous than neurons. Now, there are 6 different kinds of neuroglia. There are 4 in the central nervous system and 2 types in the peripheral nervous system. So first, in the central nervous system, we have astrocytes, we have oligodendrocytes, we have microglia, and ependymal cells. And then in the peripheral nervous system, we have Schwann cells and satellite cells. Now don't confuse this satellite cell with the satellite cells in muscle tissue. These satellite cells are neuroglia, they're not stem cells, anything like that. So this is a separate type of satellite cell from the satellite cell that we see in muscle tissue. So here we can kind of see a general picture of some neurons with their associated neuroglia surrounding them. So first off, let's look at some astrocytes. So these are kind of the blue cells here. A couple different types of astrocytes, which we'll kind of go over. But these are very star-shaped cells. They have a lot of processes, and these are going to be the largest and most numerous of all the neuroglia. So these are the astrocytes. Largest and most numerous of all the neuroglia. Now there are 2 types of astrocytes. There are here the protoplasmic. You can see a couple of those here. Protoplasmic astrocytes have many really short branching processes. They're going to be found in gray matter. Gray matter in the central nervous system. And then there is the fibrous astrocytes. We can see one right here. And fibrous astrocytes have many long, unbranched processes, and they are located actually mostly in white matter. Oops. Now these little processes of astrocytes will make contact with blood capillaries. You can kind of see them here. See that contact with the blood capillary of the astrocyte there. They also make contact with neurons. And they also make contact with the pia mater in the brain and the spinal cord. It's the connective tissue around the central nervous system. Now there are a lot of functions of astrocytes that we will very briefly go over. So this is just going to be the functions of astrocytes. So the first function is to support neurons. So they support neurons with microfilaments. It's going to be their main support that they give. Our second function is that they contribute to the blood-brain barrier. blood-brain barrier is just the abbreviated BBB. And this blood-brain barrier really maintains selective permeability into the central nervous system. So it doesn't allow just anything from the blood to cross over into the central nervous system. And the astrocytes really contribute to that barrier. Our third function is in the embryo, they secrete chemicals that regulate growth and migration and interconnection among neurons in the brain. So they'll secrete chemicals to help development. This is development of the nervous system itself, help those connections of neurons in the brain. Our fourth function is that they help to maintain the chemical environment, and this is really important for the generation of nerve impulses along the axons of neurons. Okay, and I'll put it in a different color here at the bottom, but our fifth function of astrocytes is that they play a role in learning and memory. And they do this by influencing the formation of neural synapses. So they actually help new synapses to form between neurons. Okay, so that's going to be our astrocytes. I'll kind of erase a little bit of this. I'll leave some stuff there yet. Okay, we're going to be moving on to the next neuroglia. And our next neuroglia here is the oligodendrocyte, which is this weird looking one here. These kind of resemble astrocytes, but they're going to be quite a bit smaller and they contain fewer processes. So we can see another one here. Now these processes on the oligodendrocytes are responsible for forming and maintaining the myelin sheath around central nervous system axons. So we can see this little sheath that the process of the oligodendrocyte creates there. So this myelin sheath, this is going to be a multi-layered lipid and protein covering around some axons, and it's going to insulate them and increase the speed of nerve impulse conduction. So it's a lipid and protein layer. Its function is to insulate the axons and increase nerve impulse conduction speed. Essentially, it helps that action potential to travel down the axon quicker. Okay, just a couple more cells to go over here. Next one we'll talk about are the microglial cells. So these are these little green cells here, and I'll erase this too. Okay, so microglial cells, sometimes these are also called microglia. And these are very small cells with very slender processes. They give off lots of little spine-like projections. And essentially, microglial cells function like phagocytes. So they will remove cellular debris that's formed during normal development of the nervous system. And they're also going to phagocytize or eat up and digest microbes and damaged tissue. So these guys are really important, little like trash compactors of the neuroglia in the central nervous system. The last central nervous system neuroglia we will discuss are the ependymal cells down here at the bottom. So these are going to be really cuboidal and columnar cells that are arranged in a single layer, and they have microvilli and cilia. Now these cells will line the ventricles of the brain and central canal of the spinal cord. So essentially they're going to line spaces filled with cerebral spinal fluid. So cerebral spinal fluid, abbreviated CSF. And cerebral spinal fluid is a fluid, we will talk about it a little bit later too, but it protects and nourishes the brain and spinal cord. So this fluid is very, very important. And the ependymal cells are going to line those spaces of the central nervous system that contain this important fluid. Functionally, the ependymal cells will also form the blood-brain barrier. Blood-brain barrier. They're also going to have a bigger role in the cerebrospinal fluid as well, as they will help to produce cerebrospinal fluid. They'll also help to monitor and assist the circulation of that fluid as well. So again, very important cells, very important neuroglia. In the peripheral nervous system, we find neuroglia that completely surround axons and cell bodies. Here we have Schwann cells on the left. So Schwann cells will completely encircle the peripheral nervous system axons. So they're kind of similar to oligodendrocytes, so they're going to form the myelin sheaths around axons. But where a single oligodendrocyte myelinates several axons, each Schwann cell myelinates only just one axon. A single Schwann cell can enclose as many as 20 or more unmyelinated axons, so axons that lack a myelin sheath. So these are going to be unmyelinated. And the Schwann cell is just surrounding them. It's not producing myelin or anything like that. It's just surrounding these axons. Whereas where they produce myelin, they're only going to be surrounding a single axon like we see in this picture. Now, Schwann cells are really, really important cells in the peripheral nervous system, and this is because they participate in axon regeneration. And axon regeneration, or regenerating damaged nerves, is really a lot more easily accomplished in the peripheral nervous system than in the central nervous system, and that completely has to do with Schwann cells. And we'll actually go over that in the next slide, or a couple slides from now. The other neuroglia that we have in the peripheral nervous system are satellite cells, and these are going to be very flat cells that surround the cell bodies of neurons of the peripheral nervous system ganglia. Now, besides providing structural support, these satellite cells will also regulate exchanges of materials between neuronal cell bodies and interstitial fluid. axon's surrounded by a multi-layered lipid and protein cover called a myelin sheath. These are said to be myelinated. So the sheath will actually electrically insulate the axon of a neuron and also increase the speed of nerve impulse conduction. So axons without this myelin sheath are said to be unmyelinated. Now remember, there's going to be two types of neuroglia that can actually produce myelin sheaths. In the peripheral nervous system, these are going to be Schwann cells. And in the central nervous system, these cells that can produce myelin are oligodendrocytes. Now each Schwann cell wraps about 1 millimeter around an axon's length. So if we kind of go like the length of the Schwann cell would be about 1 millimeter. And it spirals several times around the axon. Now eventually multiple layers of glial plasma membrane will actually surround the axon. So we can kind of see these layers of plasma membrane here. And the Schwann cell's cytoplasm and nucleus are going to form this outermost layer here. Now the inner portion, consisting of up to 100 layers of Schwann cell membrane, is the myelin sheath. So this could be up to 100 layers of plasma membrane. So those are going to be the inner layers. And now this outer layer, this nucleated cytoplasmic layer, the layer containing the nucleus and most of the cytoplasm of the Schwann cell, this is going to be called the neurolema. This is the outer layer. Now this neurolema is found only around the axons of the peripheral nervous system, and this is going to be the specific part that aids in regeneration of injured axons. Now there are gaps in the myelin sheath, parts of the axon that aren't covered by myelin, and these are called nodes of Ranvier. And these appear at intervals along the axon. Now remember, in the central nervous system, an oligodendrocyte can myelinate parts of several different axons. So each oligodendrocyte puts forth about 15 broad flat processes that spiral around central nervous system axons, forming a myelin sheath. But because when an oligodendrocyte myelinates, there's going to be no neurolema present. Because we have, for example, we can have this cell here, this would be an oligodendrocyte, for example, that sends out its processes to lots of different, these are going to be axons of different nerves, but the plasma membrane surrounding the nucleus and all the cytoplasm is all going to be present here. It's not around these axons. Whereas when we're looking at Schwann cells, we have the nucleus and all the cytoplasm surrounding it making up this neurolema. And that neurolema, remember, is that important part to regenerate axons. So with oligodendrocytes, there's no neurolema present because the cell body and nucleus don't envelop the axon. components of the nervous tissue are going to be grouped together in various ways. Neuronal cell bodies are often grouped together in clusters, and the axons of neurons are usually grouped together in bundles. So remember that a nerve, this is going to be a bundle of axons, and this is going to be specifically located in the peripheral nervous system. In the peripheral nervous system here. And then another term here would be tract. And tract would be a bundle of axons in the central nervous system. So same thing, but just in a different location, we call them different terms. Tracts will interconnect neurons in the spinal cord and in the brain. Now here in this picture, we kind of see this dissected or a cut brain or spinal cord. And when you see a freshly dissected brain or spinal cord, some regions will look white and glistening, while other regions appear gray. So first off, we'll discuss white matter. So white matter is primarily composed of myelinated axons. So the whitish color of myelin is really what gives white matter its name, what gives white matter its coloring. Gray matter, on the other hand, this is going to be part of the nervous system that contains neuronal cell bodies, dendrites, unmyelinated axons, even axon terminals, and neuroglia. So it kind of contains everything else. Now, gray matter appears grayish rather than white, and this is because of the Nissl bodies. Remember we kind of talked about before, these Nissl bodies? These have this kind of grayish color, and so that's where the gray matter gets its color from. There's going to be very little to no myelin within these areas. You can see in the spinal cord on the left here, we can see that the white matter surrounds an inner core of gray matter. In the spinal cord, the gray matter is kind of shaped like an H or kind of like a butterfly. You can kind of see the wings here. And in the brain, it's kind of the opposite. We get this really thin shell of gray matter surrounding this thick portion of white matter. Now remember that like muscle cells, neurons are electrically excitable. So they communicate with one another using two types of electrical signal. These are going to be action potentials or graded potentials. Now both are actually considered to be nerve action potentials. Action potentials allow for communication over short and long distances, whereas graded potentials are really just used for short distance electrical communication. Action potentials are more so used for long distance, but can also still be used for short distance. So in this picture, using the example of writing with a pen, you can see where graded and action potentials are. So the blue arrows are going to indicate graded potential, and the black arrows indicate action potentials. So here at number 1, this is where you first feel the pen, and there is a graded potential here. Maybe I'll circle it with green here. this blue graded potential develops in a sensory receptor on the skin of the finger. That graded potential is going to trigger the axon of the sensory neuron to form a nerve action potential. You can see these black arrows here indicating that nerve action potential traveling down the sensory neuron. And it's going to travel along the axon to the central nervous system, specifically to the spinal cord. Now this is ultimately going to cause the release of neurotransmitters at a synapse with an interneuron, which is actually right here at number 3. So the neurotransmitter that's released at this synapse is going to stimulate the interneuron, which is in purple here. And it's going to stimulate the interneuron in the form of a graded potential. And the interneuron is then going to produce an action potential. This is in response to the graded potential at number 3. This action potential is going to travel along the axon, which causes neurotransmitter release at another interneuron right here in the thalamus. Now, this continues as an action potential to the end of the axon to activated interneurons in the cerebral cortex. Now, once this happens, once we kind of get this action potential all the way down here to number 5, this is when perception occurs. So this is when you're actually conscious and you're able to feel the pen. So kind of all these steps from your sensory receptor in your finger all the way up. Now, if you want to write with that pen, then we kind of go the opposite way with a motor neuron. So in that case, a stimulus in the brain is going to cause a graded potential to form in the dendrites and cell body of an upper motor neuron right here. So this is an upper motor neuron. An upper motor neuron is a type of motor neuron that synapses with a lower motor neuron further down in the central nervous system in order to contact a skeletal muscle. So upper motor neurons have their cell bodies in the central nervous system. Now the graded potential, this causes an action potential in the upper motor neuron. We can see that action potential traveling down right here. And it's followed by neurotransmitter release in the spinal cord here. Now this causes a graded potential in the lower motor neuron. And this is a type of motor neuron that directly supplies the skeletal muscle fibers. So this lower motor neuron is going to go directly here. The action potential is going to travel from the spinal cord down to the muscles and directly control those muscles by a neuromuscular junction. Now, the production of graded and action potentials depends on two basic features of the plasma membrane of excitable cells. So these are the existence of a resting membrane potential and also the presence of specific types of ion channels. So these are going to be the two basic features of excitable cells, or two basic features of the plasma membrane of excitable cells that are required for producing graded and action potentials. So like most cells, the plasma membrane of excitable cells has a membrane potential. and an electrical potential difference across the membrane. So they're going to have different charges on the outside of the cell as opposed to the inside of the cell. So in excitable cells, this voltage or this electrical potential difference across the membrane is called a resting membrane potential. So let's just briefly think about a battery. So this is a battery. We have one terminal here, one terminal here. This is going to be the positive terminal, and this will be kind of the negative terminal. So let's draw a wire from the negative terminal to the positive terminal. Now electrons are going to be negatively charged, and so when you connect a wire between the positive and negative terminals, electrons, these little negatively charged particles, are going to travel along that wire. So they're going to flow from the negative terminal to the positive terminal along this wire. So this flow of charged particles is called a current. Now in living cells, we have the flow of ions rather than electrons that constitutes an electric current. So it's very similar concept, just with different particles involved. So here we're talking about the movement of ions causing this current. Now graded and action potentials, these occur because the membranes of neurons contain many different kinds of ion channels that will open and close in response to specific stimuli. So since this lipid bilayer of the plasma membrane is a really good electrical insulator, the main paths for current to flow across the membrane are only going to be through ion channels. The only way ions can travel either into or out of the cell are going to be through channels. Now when ion channels open, they allow very specific ions to move across the plasma membrane down their electrochemical gradient. Now ion channels will open and close due to the presence of gates. So this gate is going to be part of the channel protein that can seal the channel pore shut, or it can move aside to open the pore. So we can kind of see one of these gates right here. We can kind of see another little gate here. So there are actually 4 types of ion channels that allow for the electrical signals produced by neurons and muscle fibers. These are going to be leak channels. So this is a potassium leakage channel here. So leak channels is our first one. I'll probably write these here. Leak channel are ligand-gated channels. Mechanically gated channels. And voltage-gated channels. So here we have a ligand-gated channel. This is a specific ligand that's required to bind to the channel in order to open it. Mechanically gated channel, these are channels that require a mechanical stimulus, like physical movement of the plasma membrane in order to open. And then down here we have a voltage-gated channel. So this requires a very specific change in membrane potential in order to open. So first we're going to look at leakage channels. And leakage channels have gates that will randomly alternate between open and closed positions. So this is going to be very much a random thing. And that's why it's called just kind of a leak channel, because there's not a specific stimulus that's going to open it. Now, typically, The plasma membrane will have way more potassium leak channels than sodium leak channels. And also, the potassium leak channels are going to be way leakier than sodium channels. So it kind of means that the potassium channels are going to be open way more than sodium channels. Now, leak channels are found nearly in all cells and all parts of neurons as well. These are found in the dendrites, the cell bodies, and axons of all types of neurons. Then we have ligand-gated channels. Now, these are going to open and close in response to the binding of a ligand, which is another word for a chemical stimulus. Now, there's a wide variety of chemical ligands. These include, but are not limited to, neurotransmitters, hormones, or very particular ions as well. And these ligands can open or close ligand-gated channels. Now again, these are going to be very specific channels. Only a very specific ligand can bind to their binding sites to open the ligand channels. So here, this particular ligand-gated channel is specific to acetylcholine. So only acetylcholine can open this channel. Ligand-gated channels, these are going to be located in the dendrites of some sensory neurons, such as pain receptors. And they're also going to be found in the dendrites and also the cell bodies of interneurons and motor neurons. Mechanically gated channels are channels that open or close in response to mechanical stimulation, and this can be in the form of vibration, like sound waves. It could be in the form of touch or pressure or even tissue stretching. The force will actually distort the channel from its resting position, which physically opens the gate. Now, examples of mechanically gated channels are those found in auditory receptors in the ears, receptors that monitor stretching of internal organs, and also in touch receptors and pressure receptors in the skin. So we can see here the mechanically gated channel is closed. In this picture, there's going to be a physical stimulus that physically distorts the channel and opens it and allows specific ions through. So in this case, this is a sodium and calcium mechanically gated channel, so it allows those through. Then we have the voltage gated channels. And these are going to open in response to a change in membrane potential or a change in voltage. Membrane potential is another term that we use for voltage. So these channels participate in the generation and conduction of action potentials in the axons of all types of neurons. So here we can see the voltage is negative 70 millivolts inside the cell, and then so at that voltage the potassium gate is closed. When the voltage changes to negative 50 millivolts, this is when the voltage gated potassium channel will open, allowing potassium out of the cell, down its concentration gradient. Here we have a really good summary of the types of ion channels in neurons, what they do, and where they are found. So a really good summary table you can use for studying. Now, the resting membrane potential exists because of a small buildup of negative ions in the cytosol along the inside of the membrane. And we also have an equal buildup of positive ions in the extracellular fluid along the outside surface of a membrane. So we kind of look like this. So kind of we think of this as the cell, this is the extracellular fluid, and this is the cytosol inside the cell. So we kind of have this buildup of negative ions inside the cytosol here. So these are all going to be little negative ions. Sorry, the cell is closed here. And then we have this buildup of positive ions in the extracellular fluid kind of along the membrane. And this is going to be all around the cell, not just on one side like I shown here, but just think of it as all around the cell. We have this membrane potential, or this difference in electricity or difference in ions inside the cell as opposed to outside the cell. So this separation of positive and negative electrical charges, this is a form of potential energy. This is a G. So a cell that exhibits membrane potential, like we see here, this is said to be polarized. Now, resting membrane potential arises from three main factors. We have these three factors down below here. So this first one is, like I talked about on the top of this slide, is the unequal distribution of ions across the plasma membrane. So this is the unequal distribution of ions in the extracellular fluid and the cytosol. So the extracellular fluid is going to be really, really rich in sodium. So we have sodium out here, and also chloride ions are going to be out here. Sodium is going to be very abundant, and it's a positive ion. But in the cytosol, however, the main cation is going to be potassium. And there are going to be two dominant anions, which are going to be phosphates attached to molecules. So like the 3 phosphates in ATP and amino acid proteins. These are both going to be very large negatively charged anions. Now, since the plasma membrane has more potassium leak channels than sodium leak channels, remember, so the number of potassium ions that diffuse down their concentration gradient or leaving the cell is going to be much, much higher than sodium ions that diffuse across the membrane. So potassium is going to be leaving through leak channels a lot quicker and in a lot higher abundance than sodium. Now, what this does is that it causes a lot more positive ions to leave the cell than the positive ions that are entering the cell. And this additionally helps the inside of the membrane to become increasingly more negative, because we have all these positive ions that are continuously leaving. Potassium is just always leaving because there's so many leak channels. They're just leaking out of the cell essentially. And so the inside of the membrane in the cytosol becomes increasingly more negative, and the outside becomes increasingly more positive. So that's going to be our first major factor that helps with this resting membrane potential. Now, our second major factor that contributes to this resting membrane potential is the inability for anions to leave the cell. So anions, these large negatively charged ions, are not able to leave the cell. So they're all kind of stuck within that cytosol. Most anions cannot follow potassium out of the cell, and this is because they're attached to non-diffusible molecules like ATP or really large proteins. So they're stuck inside the cell. Then our third major factor contributing to this resting membrane potential is the nature of the sodium-potassium ATPase pumps. So membrane permeability to sodium is going to be very low, and this is because there are very few sodium leak channels, but they still do diffuse into the cell every now and then. If left unchecked, this really slow inward leakage of sodium into the cell would eventually destroy this resting membrane potential. So we kind of slowly have sodium kind of leaking in slowly over time, and it just kind of builds up over time. And then we would no longer have this difference in potential across the membrane. So this small inward and outward leak of sodium and potassium is going to be offset by these sodium-potassium ATPase pumps. These pumps help to maintain the resting membrane potential by pumping out sodium as fast as it leaks in, and at the same time it brings in potassium. So we have these pumps here. This is going to be a sodium-potassium ATPase pump. And so we're going to take the sodium that leaks in and we're going to pump that back out. And we're also going to pump in some of this potassium that leaks out. And remember, this pump uses ATP. So this pump, remember, is going to expel 3 sodium for every 2 potassium that they import. So this pump works to pump 3 sodium out. For every 2 potassium, it pumps back into the cell. So it's really helping to maintain that resting membrane potential. Now the resting membrane potential, this can be measured by putting the tip of a recording microelectrode inside the cell and having a reference electrode just placed outside the cell within the extracellular fluid. Electrodes, these are going to be devices that conduct electrical charges. Sorry, this would be the electrodes themselves. So this electrode and this electrode. Now the recording electrode and the reference electrode are connected to an instrument known as a voltmeter. And the voltmeter is going to be the actual device that detects the electrical differences between the 2 electrodes. So it's actually going to detect the electrical differences across the plasma membrane. Now in neurons, the resting membrane potential usually ranges between -30 to -90, but for our class, a typical value is going to be -70 millivolts. That's going to be a typical resting membrane potential in neurons. So in this picture, we can visually see the 3 main factors that affect the resting membrane potential. So we can see resting membrane potential is just labeled here. You can see this negative charge on the cytosol side of the membrane and this positive charge buildup on the extracellular fluid side of the membrane. So the leak channels of sodium and potassium are here. Remember that there's going to be a lot more potassium than sodium leak channels in the membrane. We can see all these anions that are here that are stuck inside the cytoplasm, inside the cell. You can see all these, the anions, the proteins, and the phosphate ions that really contribute a lot to that negative charge inside the cytosol. And then there's also going to be the sodium-potassium ATPase pump. Remember, this requires ATP to pump 3 sodium out of the cell for every 2 potassium they pump back into the cell. Now we're going to move on to graded potentials. So a graded potential is a small deviation from the resting membrane potential, and this is going to either make the membrane more polarized, which is going to essentially make the inside more negative. We can see in this picture up here. Or a graded potential could also make the membrane less polarized or make the inside a little bit less negative. Now, when the response makes the membrane more polarized, it's termed a hyperpolarizing graded potential. So we can see here the resting membrane potential is at about -70. And we have this graded potential that brings the resting membrane potential a little bit more negative, dropping it down to maybe 72, kind of about right here, maybe 73. So this is a hyperpolarizing graded potential. It makes the membrane more polarized, bringing the membrane further into the negative, makes the inside more negative. Now, when the response makes the membrane less polarized, as we can see on this bottom graph here, it is termed a depolarizing graded potential. So here again, we can see that resting membrane potential about -70. And as we get less negative inside the cell, this is a depolarizing action. So we kind of brought that membrane up to -60 so that the inside of the membrane becomes less negative. Now, a graded potential occurs when a stimulus causes a mechanically gated or ligand-gated channels to open or close in an excitable cell's plasma membrane. Remember, our excitable cells, these are going to be neurons and muscle cells. Well, we're mainly going to talk about graded potentials in terms of neurons. Now, typically, the mechanical and ligand-gated ion channels are found in the dendrites of sensory neurons, and then ligand-gated channels are a lot more numerous in the dendrites and cell bodies of interneurons and motor neurons. So this one up here is going to be both mechanically gated and the ligand gated. And we're going to see the ligand gated channels being much more numerous in just the dendrites and cell bodies of interneurons and motor neurons. So having this type of information, we know that graded potentials will occur mainly in the dendrites and the cell bodies of neurons. So we don't really see graded potentials happening in muscle cells. Even though muscle cells are excitable cells, we mostly just see them happening in the dendrites and in the cell bodies of neurons. So in this picture here, we can see a mechanically gated channel that will allow calcium, and it looks like sodium through when it's open, when applied pressure comes into play. And this is a depolarizing graded potential. And here we have a ligand-gated channel that requires acetylcholine to bind to it. And when acetylcholine does bind, it will allow sodium and calcium into the cell, and it will allow potassium out of the cell. And this is also a depolarizing graded potential. And another ligand-gated channel, which requires the binding of glycine, so that when glycine binds, it allows chloride into the cell, making the inside of the cell more negative, or a hyperpolarizing graded potential. To say that the potentials are graded means that they vary in amplitude. And this is depending on the strength of the stimulus. So the potential will be larger or smaller depending on how many ligand-gated or mechanically gated channels have opened or closed and how long each remains open for. So the opening and closing of these ion channels alters the flow of specific ions across the membrane, producing a flow of current that is localized, which means that it spreads to adjacent regions along the plasma membrane in either direction from the stimulus source for a short distance, and then it gradually dies out as the charges are lost through the membrane leak channels. So this is kind of a mode of travel by which graded potentials will die out as they spread along the membrane. And this is called decremental conduction. So we'll talk about this graph below in just a second, but imagine we have an axon from a membrane right here. And if there's a stimulus right here, we're going to have the membrane potential be strongest at this area, and it'll spread to adjacent areas but be smaller as it gets further away from the stimulus source. So that the potential is going to die out as it spreads along the membrane. So this is the exact definition of decremental conduction, and this happens with graded potentials. So they're going to die out within a few millimeters, so because of this, graded potentials are really only useful for short distances. So in this graph on the bottom here, you can see that membrane potential changes are going to be very dependent on the strength of the stimulus. So we see the strength of the stimulus on this bottom graph here. So stimulus 1 is going to be the lowest strength, producing the smallest graded potential. Stimulus 2 is a little bit stronger, producing a little bit stronger of a depolarizing membrane potential change, or a depolarizing graded potential. And then stimulus 3 is the strongest in this example, showing the strongest depolarizing graded potential here. Now, although an individual graded potential undergoes decremental conduction, it can become stronger and last longer by summating with other graded potentials. So here we have this summation. Now summation, this is going to be the process where graded potentials in this case are going to be added together. So if two depolarizing graded potentials will summate, the net result is a larger depolarizing potential. So we could actually see this with this graph up above here. So we have stimulus 1, which is a depolarizing stimulus, which causes a depolarizing graded potential. And if it was just that stimulus, it would continue down and die out over time and over distance as well. But if we get a second stimulus shortly after the first here, we get this first stimulus causing a depolarizing potential, and then we get this second depolarizing stimulus, which is going to be right here as this first stimulus is dying off. We get as the first graded potential, is dying off, we get a second stimulus, which causes an even stronger depolarizing graded potential. And then we come back down to the resting membrane potential. The same is going to happen if two hyperpolarizing potentials summate. Then the net result is a larger hyperpolarizing graded potential. And then if we have two equal but opposite graded potentials, if they are going to summate, so one depolarizing and one hyperpolarizing, they're going to cancel each other out, and then the net result is that the graded potential disappears. Moving on to action potential. So remember, there's a difference between graded potentials and action potentials. So an action potential is a sequence of rapidly occurring events that decreases and reverses the membrane potential, and then eventually restores it back to its resting state. So this is going to be back to the resting membrane potential. Now the action potential has two main phases. There's going to be a depolarizing phase, and a repolarizing phase. So these are the two main phases. There's a couple other little smaller phases which we will discuss yet. But during the depolarizing phase, the negative membrane potential becomes less negative. It's actually going to reach 0 and become positive. We'll see this in a graph in the next slide. And then during the repolarizing phase, the membrane potential is restored back to the resting state of negative 70 millivolts. So an action potential occurs in the membrane of an axon of a neuron when depolarizing reaches a certain level. So this certain level is called threshold. And this depolarizing level, this threshold, is negative 55 millivolts in most neurons. So once we kind of have enough stimulus to bring depolarization to threshold, this sets in motion these sets of events that brings about an action potential. So here we have this stimulus here, and it's going to bring the membrane potential up to negative 55 millivolts, which is going to cause the action potential and initiate the depolarizing phase. So once the membrane, during this depolarizing phase, once the membrane reaches a peak here at about positive 30 millivolts, this is when it begins the repolarizing phase. And then following the repolarizing phase, there's also a after hyperpolarizing phase. And then this is where the membrane potential temporarily becomes more negative than the resting level of negative 70. Now we're going to go over each of these steps, but this is just kind of the overview of what happens during an action potential. So this figure here really is a great one that shows what ion channels are open during each phase of an action potential. So during our regular resting membrane potential here, in kind of the purple color gray right here, this is when all of our voltage gated channels are closed. So voltage gated sodium channels, they're in this resting state, which is closed, and voltage gated potassium channels are going to be closed. Now, after there's a stimulus and the cell reaches threshold, then we reach this green color here. This is when our voltage-gated sodium channels are activated and their activation gates are open. So sodium is going to rush into the cell, and this brings the cell potential very quickly positive. So this is during that depolarizing phase. So sodium rushes into the cell, and all those positive ions bring the interior of the cell all the way up to positive 30. Now, after a certain time, the voltage-gated sodium channels will deactivate. So we'll kind of talk about that yet, but they deactivate or inactivate, another term for that. And this is when the voltage-gated potassium channels open. So that's during this little cream phase here. One thing to note that is at this point, at threshold, all voltage-gated channels in the area are opened. But the voltage-gated sodium channels open and close so much faster than voltage-gated potassium channels. So voltage-gated sodium channels, they very quickly open and very quickly close, so they're going to be really fast channels. Whereas the potassium channels, these are going to be very slow, especially compared to the sodium channels. So in this repolarizing phase, the potassium channels are finally opened and the potassium starts to leave the cell. So then we kind of have that drop in a positive charge within the cell because those positively charged potassium ions are leaving. Now, because these potassium ion channels are so slow to close, they kind of stay opened a little bit longer than after getting to the resting membrane potential. So this allows more potassium ions to leave the cell, and it causes the cell membrane to go more negative than that resting membrane potential. And then they kind of close completely, and with the help of the sodium-potassium ATPase pump, the cell gets back to that resting membrane potential. Now, this generation of an action potential depends on whether a certain stimulus is able to bring the membrane potential to threshold. So this threshold potential is really, really important. An action potential will not occur in response to a subthreshold stimulus. This is going to be a very weak depolarization that can't actually bring the membrane all the way up to threshold. However, an action potential will occur in response to a threshold stimulus. So this is a stimulus that is just strong enough to depolarize the membrane all the way to threshold. Now, in response to a suprathreshold stimulus, we're going to get several action potentials. So we can see all these action potentials that come. So a suprathreshold stimulus is a stimulus that's strong enough to depolarize the membrane well above the threshold. So this figure here shows the steps of an action potential in regards to the ion channels. So we have the sodium channel in green and potassium channels in yellow. So as you can kind of see in these pictures, the voltage-gated sodium channels are a bit different from the potassium channels. And this is because they have two different gates. They kind of have an activation gate and an inactivation gate. Sometimes I call this inactivation gate kind of a plug because it kind of plugs up the bottom of the ion channel. So here at the resting state, both the sodium and potassium voltage-gated channels are closed. You can see we have this activation gate on the sodium channel. This is closed, so nothing is getting through. And this bottom gate on the potassium channel is also closed. Now, once there's a stimulus to the cell that brings the membrane to threshold, this is when we reach that depolarizing phase. So this is where the voltage-gated sodium channels open. So we can see it's open here now. And sodium is going to rush into the cell, as we can see here. This is going to increase the positive charges or the buildup of positive charges inside the membrane, and the membrane becomes depolarized. It's at this point that technically these potassium channels are starting to open as well, but like I said, they're really slow, so they're just like barely starting to open yet. They're just really slow to open. Now, as soon as the sodium channels are opened, they start to inactivate. This plug starts to close the sodium channel as soon as they open. And this inactivation gate completely plugs up the sodium channel once the cell reaches positive 30 millivolts. This is where we get that repolarizing phase. So during this repolarizing phase, the sodium channels are still technically open. You can see this activation gate is still technically open, but they are deactivated. We have this plug that plugs up the bottom of the sodium channel that still does not allow anything through. Now at the same time, the voltage-gated potassium channels are now finally opened, as we can see here. And some potassium is going to leave the cell. And a few negative ions will build up on the inferior of the cell. So we're bringing that potential back down from that super high positive. So now throughout this repolarization phase, then potassium will continue to flow out of the cell, and this will eventually restore the resting membrane potential. So during this phase, the sodium channels also will close completely. So this gate will close during kind of the end of the repolarization phase. And once that gate closes, then this inactivation gate or inactivation plug will also be removed. Now, once all the potassium gates are closed, then the resting state is returned back to normal, and we return back to the initial state. So looking back at this figure again, there are two terms that we haven't talked about. These are the absolute refractory period and the relative refractory period. So first off, the absolute refractory period, this is the point of an action potential that another action potential cannot fire, no matter how large the stimulation. So this absolute refractory period is really due to the voltage-gated sodium channels being inactive. So having that inactive the inactivation plug being in place, and so they're completely unable to open again during that time. Then we have that relative refractory period, and this is when another action potential can fire during this time, but only if the stimulation is large. So one of those suprathreshold stimuli. So during this relative refractory period time, so kind of from like here to here, this is when most of the sodium channels in the area are still inactive and the potassium channels are still closing, so it's really difficult to have another action potential fire. Here, I didn't really mark where the absolute refractory period is in this time, but it's kind of from here till here. This is when another action potential cannot fire at all. Here we have a nice table, be really useful for studying, and it shows a really good comparison between the graded potentials and action potentials. So again, everything that we've gone over before, types of channels, their duration, how they're conducted, that kind of thing. So all stuff that you can use to study the differences between graded and action potentials. So now that we know a little bit about action potentials, we need to know how they move along the neuron axon. Now there are two ways that action potential moves along the axon. These are either by continuous conduction or saltatory conduction. So the first thing you need to know is that action potentials need to fire at every available space on an axon in order to continue moving. So in other words, the voltage-gated channels have to open in one area before they can activate the next ones to open, and so they move along the action, or they move the action potential down the axon that way. So if there's available space, action potential is going to fire here as those voltage-gated channels open, and then here, and then here, and then here. And so every available voltage-gated channel will have to open in order to propagate or move that action potential down the axon. So first, in continuous conduction on this left picture here, there's going to be a step-by-step depolarization and repolarization of each adjacent segment of the plasma membrane. So because every single channel has to open, in a few milliseconds, the action potential doesn't move very far. So we can see here at 1 millisecond, it's right here. At 5 milliseconds, it hasn't gone very far, it's right here. And then at 10 milliseconds, we have yet to travel very far at all. And so every single voltage-gated channel has to open along the membrane, so it takes a long time in continuous conduction for the action potential to propagate down the entire length of the axon. Now, action potentials will propagate more rapidly along myelinated axons than along unmyelinated axons. So this is because myelinated axons will use what is called saltatory conduction. So saltatory conduction is a special mode of action potential propagation that occurs along myelinated axons, and this is because of the uneven distribution of voltage-gated channels. Wherever there's myelin, as we can kind of see in this picture, there's going to be myelin in this area, the channels are going to be covered. They're completely covered by that insulating myelin, and so there doesn't have to be an action potential propagated there because they're covered. In saltatory conduction, the ionic currents will go through the cytosol from one node of Ranvier to the next. So in this way, saltatory conduction can propagate much further, much faster. So you can see at 1 millisecond, we're here at that axon hillock. At 5 milliseconds, the action potential will jump to the next node of Ranvier. So it's kind of called jumping conduction, is another term for saltatory conduction, but it's kind of a jumping motion, where at 10 milliseconds, it's jumped to this node and then again jumped to that node. So it's much faster than saltatory conduction. Because saltatory conduction, we're only here on the axon, whereas on the same axon, if it was myelinated, with saltatory conduction, the same time frame, it's about here. So this much distance difference between continuous conduction and saltatory conduction. So it's quite a big difference. Now there are a couple of factors that affect propagation speed. So this is in continuous and in saltatory conduction. So first off is axon diameter. So a larger, thick diametered axon will propagate action potentials faster than smaller ones, and this is due to their larger surface area. So we're going to get an increased propagation speed with thicker diametered axons. The amount of myelination also affects propagation speed greatly because, like we just learned, action potentials will propagate more rapidly along myelinated axons than unmyelinated ones. So if we have more myelin, this is going to be an increase in propagation speed. Then lastly is temperature. So at lower temperatures, axons will propagate action potentials much more slowly. Everything's kind of more slowly at low temperatures. So with an increase in temperature, this will also increase propagation speed. Now recall back to when we talked about muscles and neuromuscular junctions, that a synapse, this is where communication occurs. So I'll put this here. So remember, this is where communication occurs. Now in this chapter, we're going to talk about communication between two different neurons instead of before when we talked about communication between a neuron and a muscle. So between two different neurons. So first off, we have to kind of cover a couple different terms. So first off, we're going to talk about the presynaptic neuron. And this refers to a nerve cell that carries a nerve impulse towards the synapse. Then we also have the postsynaptic neuron. And these are the cells that receive the signal, and then they carry the nerve impulse away from the synapse. not supposed to have an either. Okay, so those are the presynaptic neuron and the postsynaptic neuron. So these are going to be the two different neurons that are communicating at a synapse. So there are two different types of synapse that we can discuss too. The first one here is an electrical synapse. So at electrical synapses, the action potentials conduct directly between plasma membranes of adjacent neurons, and they communicate through these structures that we already learned about. These are called gap junctions. So they're actually going to physically connect these cells. Remember before we saw that there was a synaptic cleft or a space between the two communicating cells. But with electrical synapses between two neurons, there's actually no space. The gap junctions will physically connect the presynaptic neuron with the postsynaptic neuron. Now, electrical synapses will have two main advantages. They provide faster communication. And this is because we don't have to wait for the neurotransmitters to cross over that gap, cross over the cleft, and then bind to receptors on the postsynaptic neuron. That'll take some time, whereas the gap junctions is kind of in this instantaneous communication. So they allow for faster communication, and because of that, they allow for synchronization. They can allow an electrical signal to essentially pass through multiple neurons at almost instantaneous speeds. Then we have the chemical synapse, ones that we're more familiar with. This is when the pre- and postsynaptic neuron are really close to one another, but they don't actually touch. They're going to be separated by that synaptic cleft, and the synaptic cleft is filled with interstitial fluid. Now, the time required for the action potential to come to the synaptic cleft, then for the neurotransmitters to cross the cleft, and then for the action potential to fire in the postsynaptic neuron, this is all referred to as synaptic delay. So this synaptic delay we see in the chemical synapse because of the time it takes these chemicals to move, but we don't see a synaptic delay in electrical synapses. And so chemical synapses will be much slower than electrical synapses. Most synapses between neurons are axodendritic. Now this is where the axon from one neuron sends the action potential directly to the dendrites of another neuron. There are going to be some other synapses that are going to be axosomatic. So this is from axon to cell body, so you can see here. Or there's a couple others that will be axo-axonic, where we have the axon going directly to the axon of another neuron. Now, in addition to these kind of physical differences, the synapses could all either be electrical or chemical. So lots of different classifications we could have here. So here we have a chemical synapse. So this is going to be a axodendritic. As we can see in this little picture here, we have the axon of our presynaptic neuron having a synapse with the dendrites of a postsynaptic neuron here. So this is a chemical synapse where the action potential has reached the axon terminal in the presynaptic neuron. So here we have the axon terminal of the presynaptic neuron, and the nerve impulse has reached the end bulb here. So here at number 2, we see that the depolarizing phase of the nerve impulse has opened these voltage-gated calcium channels that are present in the membrane of synaptic end bulbs, which is going to be labeled here. Now, because calcium ions are going to be more concentrated in the extracellular fluid, calcium rushes into the cell. So we can see all these calcium, these little purple dots here, they're going to be rushing into the cell there. Now, an increase in calcium ions inside the presynaptic neuron serves as a signal that's going to trigger exocytosis of synaptic vesicles. Now, these vesicles are filled with neurotransmitters that are going to be released into the synaptic cleft because of this extra calcium in the end bulb. So those calcium, they trigger exocytosis here at 3, and these red neurotransmitters, which are going to be, most likely they're going to be acetylcholine in this case, they are going to be released into the synaptic cleft here. Now those neurotransmitters will diffuse across the synaptic cleft, and they will bind to these neurotransmitter receptors. These are going to be in the postsynaptic neuron's plasma membrane. They're going to bind to these ligand-gated channels, which when they open, as you can see here, they're going to allow specific ions to cross the membrane into the postsynaptic neuron. For example, here we can see sodium is coming in through these ligand-gated channels when the neurotransmitters bind to the receptors. So as those ions flow through the open channels, the voltage across the membrane changes. This is going to be the voltage across the postsynaptic neuron membrane. So this change in the membrane voltage, we call it the postsynaptic potential. And a postsynaptic potential, it can be depolarizing or it can be also hyperpolarizing. Here, I'll do this arrow a bit better here, but this is hyperpolarizing. So in this example here, we see sodium ions rushing into the postsynaptic neuron when the neurotransmitters bind to the receptors on the postsynaptic neuron membrane. So the opening of these sodium channels allows the influx of sodium, which causes depolarization, right? Because we're going to be sodium rushing into the cell is going to increase the potential, bring the potential further away from the negative, bring it more and more positive because sodium is a positive ion, and it's going to cause depolarization, which will cause a nerve impulse if the depolarization reaches threshold. However, there are other cases where these ligand-gated channels are going to be chloride channels, or they may be potassium channels. And if either of these open and either allow chloride into the postsynaptic neuron or allow potassium out, this would cause hyperpolarization. So in these cases, remember chloride will move into the cell to bring it more negative, and potassium ions will leave the cell, also resulting in making the postsynaptic neuron more negative. Now, when the depolarizing postsynaptic potential reaches threshold, it's going to trigger that action potential in the axon of the postsynaptic neuron, like we saw here. Now, at most chemical synapses, only one-way information transfer can occur, just from presynaptic to postsynaptic. A neurotransmitter causes either an excitatory or an inhibitory graded potential. So a neurotransmitter that causes depolarization of the postsynaptic membrane is excitatory because it brings the membrane closer to threshold. So a depolarizing postsynaptic potential is called an excitatory postsynaptic potential. These are also called EPSPs. Although a single EPSP normally doesn't initiate a nerve impulse, a postsynaptic cell does become more excitable. Now, a neurotransmitter that causes hyperpolarization of the postsynaptic membrane is inhibitory. And this is because during hyperpolarization, generation of an action potential is more difficult than usual. And this is because the membrane potential inside becomes more negative. And so it's even further from threshold than when it was at resting membrane potential. So a hyperpolarizing postsynaptic potential is termed an inhibitory postsynaptic potential, also abbreviated IPSP. And a postsynaptic neuron can receive many signals at once, both EPSPs and IPSPs. And we'll go over those yet. Each neurotransmitter receptor has one or more neurotransmitter binding sites to which a specific ion can bind to. When a neurotransmitter binds to the correct neurotransmitter receptor, an ion channel opens and a postsynaptic potential forms, either an IPSP or an EPSP, and this postsynaptic potential is going to form in the membrane of the postsynaptic cell. Now there are kind of two types of neurotransmitter receptors that we're going to talk about, ionotropic receptors and metabotropic receptors. Ionotropic receptors, these are types of neurotransmitter receptors that contain a neurotransmitter binding site and an ion channel in the same receptor. So these top two are going to be types of ionotropic receptors, the purple one and the orange one here. So in other words, the neurotransmitter binding site and the ion channel are components of the same protein. So you can see, well, you can see it better here in this orange one where we have the receptor or the binding site for neurotransmitters here, and the ion channel is through the middle. So these are both parts of the same protein. Now, many excitatory neurotransmitters will bind to ionotropic receptors that contain cation channels. Remember that EPSPs will result from opening these ion channels. So our top one here is going to be the excitatory postsynaptic potential. This is where we have an ionotropic acetylcholine receptor that has a cation channel. This is going to be the most common type of excitatory receptor. So when acetylcholine binds, it allows calcium and sodium into the cell, and it allows potassium out of the cell. So this is an excitatory, or an EPSP. We also do sometimes see IPSPs resulting from ionotropic receptors. An example would be a GABA receptor. So this is GABA. This is a inhibitory neurotransmitter. So when GABA binds, it allows chloride into the cell, resulting in an IPSP. Down here, we have the metabotropic receptor. This is a type of neurotransmitter receptor that contains a neurotransmitter binding site, but it lacks an ion channel as part of its structure. However, a metabotropic receptor is coupled to a separate ion channel by a type of membrane protein called a G protein. So here we can see this left receptor being a metabotropic acetylcholine receptor, and we can see that it just has a binding site here. It's just a receptor with a binding site, and that is all. So when acetylcholine binds, it sends communication to this little blue kidney bean shaped G protein. The G protein will travel over to this ion channel, and it will activate the ion channel, opening this potassium channel in this case. So these are two separate proteins as part of a metabotropic receptor. So when a neurotransmitter binds to the metabotropic receptor, the G protein will either directly open or close the ion channel, or it may act indirectly by activating another molecule, a second messenger in the cytosol, which in turn will then open or close the ion channel. Some inhibitory neurotransmitters will bind to metabotropic receptors. These are mostly going to be ones that are linked to potassium channels. So when acetylcholine binds to the metabotropic receptor, it will open the potassium channel, allowing potassium out, resulting in an IPSP. Removal of the neurotransmitter from the synaptic cleft is really essential for normal synaptic function. So if the neurotransmitter could linger in the synaptic cleft, it would influence the postsynaptic neuron or muscle fiber or gland indefinitely. So after a neurotransmitter has had its single effect on the receptor, neurotransmitters are removed from the cleft, and they're removed from the cleft in one of three ways. The first way would be by diffusion. So some of the neurotransmitter molecules just kind of diffuse away from the synaptic cleft. And once it's out of reach of its receptors, it can no longer exert any effect. It can also be degraded by an enzyme, or enzymatic degradation. So certain neurotransmitters are inactivated through enzymatic degradation. An example of this would be acetylcholinesterase. Acetylcholinesterase breaks down acetylcholine in the synaptic cleft. It could also be taken up into the cells. So many neurotransmitters are actively transported back into the neuron that released them. So back into the presynaptic cleft, or presynaptic neuron, sorry. And other neurotransmitters, they can be transported into neighboring neuroglia, which will also take up neurotransmitters. A typical neuron in the central nervous system receives input from 1,000 to 10,000 different synapses. So integration of these inputs involves summation of the postsynaptic potentials that form in the postsynaptic neuron. Recall that summation is the process by which graded potentials are added together. Now, summation can be spatial or it could be temporal. So first off, we'll talk about temporal summation. So temporal summation is going to be the summation of postsynaptic potentials in response to stimuli that occur at the same location in the membrane of the postsynaptic cell, but it occurs at different times. So for example, temporal summation results from the buildup of neurotransmitter released by a single presynaptic end bulb 2 or more times in rapid succession. So here we would get temporal summation with 4 different impulses. Then we have spatial summation. This is the summation of postsynaptic potentials in response to stimuli that occur at different locations in the membrane of a postsynaptic cell at the same time. So for example, we would get like one synapse here and one synapse here, and they're going to have postsynaptic potentials or resulting postsynaptic potentials that occur at the same time. So spatial summation results from the buildup of neurotransmitter released simultaneously by several presynaptic end bulbs. So like we get here. The sum of all the excitatory and inhibitory effects at any given time determines the effect on the postsynaptic neuron. So here we have several presynaptic neurons all around this one postsynaptic neuron. And so there are lots of EPSPs and IPSPs all coming in to this postsynaptic neuron. And this postsynaptic neuron at the trigger zone is going to summate all of these EPSPs and IPSPs and really kind of determine whether an action potential needs to be generated. So it's going to summate everything and it's going to determine the response, and it could have one of 3 responses. So the first total response could be EPSP. So this is where the total excitatory effects are going to be greater than the total inhibitory effects, but this is going to be still less than threshold. So it's just an excitatory graded potential at this point. The second response that this postsynaptic neuron could have would be a nerve impulse or an action potential. So this is where the total excitatory effects are greater than the total inhibitory effects, and also threshold is reached. So this results in an action potential being generated. The third response that this postsynaptic potential could have would be an IPSP. So this is where the total inhibitory effects are greater than the total excitatory effects, and the membrane is going to hyperpolarize. So it goes further from the resting membrane potential. About 100 substances are either known or suspected to be neurotransmitters. And some neurotransmitters will bind to their receptors and act really quickly to open or close ion channels in the membrane. And remember, the result can always either be excitation or inhibition of the postsynaptic neurons. This could be dependent on the neurotransmitter and also dependent on the receptor type. Now, many neurotransmitters are also hormones that are released into the bloodstream by endocrine cells in organs throughout the body. Within the brain, there are going to be certain neurons that are called neurosecretory cells that secrete neurotransmitters, but also secrete hormones. So here's some small molecule neurotransmitters that we're briefly going to go over. So some neurotransmitters that we know of are acetylcholine, amino acids, biogenic amines, ATP, and some other purines also can act as neurotransmitters, as well as nitric oxide and carbon monoxide. So here are some of the most common neurotransmitters. First one we have here is acetylcholine. This is probably the best studied neurotransmitter, and it's released by many peripheral nervous system neurons and some central nervous system neurons. Draw a line over here. It's going to be an excitatory neurotransmitter at some synapses. So like the neuromuscular junction where it binds to ionotropic receptors, it's going to be excitatory in this case. But it also does act as an inhibitory neurotransmitter at other synapses. So this would be an example of this would be in the heart where it binds to metabotropic receptors. And then in the neuromuscular junction, remember that's ionotropic. Our next neurotransmitter would be our amino acids. So there are several amino acids that act as neurotransmitters in the central nervous system. Ones like glutamate and aspartate, these have really good, really powerful excitatory effects. And then we have down here gamma aminobutyric acid, also known as GABA, and glycine. These are both really important inhibitory neurotransmitters. GABA, remember, is going to be a really important neurotransmitter that binds to ionotropic calcium receptors. Then on the right here, we have the biogenic amines. These are going to be modified amino acids. So things like norepinephrine, this plays a role in arousal, which is awakening from deep sleep. It also has roles in dreaming and regulating mood. A smaller number of neurons in the brain uses epinephrine as a neurotransmitter. Both norepinephrine and epinephrine are used as hormones as well in the body, and we will learn a little bit more about that later. Neurons containing dopamine, these are active during emotional responses as well as addictive behaviors and pleasurable experiences. And then serotonin here is concentrated in the neurons that are in part of the brain called the RAF nucleus, which we will also talk about later. So serotonin is really useful in the RAF nucleus in the brain. And it's thought to be used in sensory perception, temperature regulation, even control of mood, appetite, and induction of sleep. Those are going to be the main neurotransmitters that we will discuss for this class. You can see nitric oxide, carbon monoxide. These are also neurotransmitters, as well as purines and neuropeptides, though those are less important for our purposes. Now, one of the final things that we will discuss in this chapter is going to be neural circuits. So the central nervous system contains billions of neurons that are organized into these really complicated networks, and these complicated networks are called neural circuits. These are going to be functional groups of neurons that process specific types of information. So we have that definition here at the top. Now there are several different types of circuits that we will go over, including a simple series, diverging, converging, reverberating, and parallel after discharge. So in a simple series circuit, we're just going to have a presynaptic neuron stimulates a single postsynaptic neuron. The second neuron will then stimulate another, and so on. So just for an example, we're just going to get like this presynaptic neuron. This is going to be the axon. This is the axon terminal, which connects to the dendrites on another neuron. I'm just not drawing the dendrites, but connects to the cell body. So this is just a simple series circuit where we have our first, this is going to be our presynaptic neuron for this synapse, connects to our postsynaptic neuron here. Then you have another synapse and so on and so forth. So it's just one connected to one connected to one. However, most neural circuits are going to be a lot more complex than this. So a single presynaptic neuron may synapse with several postsynaptic neurons. This type of arrangement is called divergence. And so we would name this a diverging circuit, where we have one presynaptic neuron that is synapsing with 2 postsynaptic neurons here. So this allows one presynaptic neuron to influence several postsynaptic neurons, or several muscle fibers, or several glands, for example. In a diverging circuit, the nerve impulse from this single presynaptic neuron causes the stimulation of increasing numbers of cells along the circuit. So here we just go from 1 cell to 2 cells to 4 cells. So we're increasing the number of cells that are influenced, or influencing the number of cells that receive the nerve impulse. So that's that diverging circuit. Another type of arrangement called convergence, this is where several presynaptic neurons will synapse with a single postsynaptic neuron. So this arrangement allows for more effective stimulation or inhibition of the postsynaptic neuron. So we call this one a converging circuit here. This is where the postsynaptic neuron, this orange one down here, this receives nerve impulses from several different sources. In this example, we have 3 different sources providing input to that single postsynaptic neuron. Some circuits are also organized so that stimulation of the presynaptic cell causes the postsynaptic cell to transmit a series of nerve impulses. So a circuit like this would be called a reverberating circuit. So in this pattern, the incoming impulse stimulates, so the incoming impulse up here is going to stimulate the first neuron, which stimulates the second, which stimulates the third, and so on and so forth. But there are branches from alterneurons that synapse with earlier ones. So you can see here, this branch goes here, and then we're going to branch out and stimulate earlier neurons in that circuit. So this arrangement sends impulses back through the circuit again and again. So among the body responses that are thought to be the results of output signals from reverberating circuits are things like breathing, or coordinated muscular activities like walking, short-term memory even. These are all thought to be a result of a reverberating circuit. Then our last type of circuit is a parallel after discharge circuit. So in this type of circuit, we have a single presynaptic cell up here, and this is going to stimulate a group of neurons, each with synapses to a common postsynaptic cell. So this is going to be our first cell here, and we see that its axon stimulates here, this cell, this cell, this cell, and then those three synapses will all come back down to a single postsynaptic cell. So these parallel after discharge circuits may be involved with really precise activities, things like making mathematical decisions. So here's another term that we need to know, and that is plasticity. So plasticity is the capability to change based on experience. So this is how your nervous system changes to create new connections in your brain. These new connections between neurons are how your brain learns and how you make new memories. So despite the plasticity of nervous tissue, mammalian neurons really have limited powers of regeneration. So regeneration is going to be the capability to replicate or repair themselves. So neurogenesis is the birth of new neurons from undifferentiated stem cells, and this occurs regularly in animals. However, in the adult central nervous system, there is a significantly lowered, if not like no repair, due to a couple of factors. First off, the oligodendrocytes. These are found in the brain. Remember those neuroglia in the brain that produce myelin? They have inhibitory factors. And the oligodendrocytes have these inhibitory factors that really keep neurons from regenerating. There also aren't any growth stimulating cues in the adult central nervous system. And also, after axonal damage, the nearby astrocytes will proliferate really rapidly, forming a type of scar tissue. And this acts as a physical barrier for regeneration. This is due to astrocytes. So astrocytes kind of grow, or they divide really quickly, and they produce kind of this scar tissue that physically prevents regeneration of neurons. So it's really too bad we can't actually have regeneration of neurons within our central nervous system.